This short video, I want to try to describe the current hypothesis or the current idea for muscle contraction. So these are some great figures and pages out of chapter 11 out of our textbook. I've copied some figures online that I just find that are interesting, and I'll try to share these with you. Some of these notes I'll ask you to review on your own, but I'll try to review muscle contraction. So know the basic muscle fiber or muscle cell structure. We've gone over that previously in another short video. So those are some nice figures at the beginning of chapter 11. But what we want to talk about now is muscle contraction. So this is called the sliding filament model or sliding filament hypothesis for muscle contraction. This is an awesome picture. I copied it online or from another textbook, I think. A similar picture that we have out of our textbook is figure 1123, which is on page 351. And actually, you should look at all these different figures, so 1119 up to 1123. And they're all pretty good figures that describe muscle contraction. So probably you remember this from an anatomy and physiology class, but the bottom part of this panel, or this picture, is the thick filament. The thick filament is made up of only myosin protein. So this is all myosin. Myosin is a large protein, and it has a neat structure that it has, that it has a myosin tail. So this would be described or drawn as a myosin tail. But then it also has a myosin head. So here's a globular portion, or sort of a ball, that's considered the myosin head. And the myosin head is the part of the myosin protein that needs to bind strongly to the actin protein. So somehow we need to get this strong binding to occur, so a cross bridge formation will occur, so we can, as long as ATP is provided, we can have muscle contraction and relaxation. So how do we get this strong cross-bridge formation? So this is the thick filament on the bottom. The thin filament on top, it is smaller in size, yet it's made up of actually three main proteins. We've all heard of actin, so these circular balls represent actin, and they're sort of in a helical twisted structure. So there's actin. The thin filament is also made up of another protein called troponin, and that's drawn here as this like oval-shaped thing. And another protein called tropomyosin. And tropomyosin is drawn as this thin line that actually sort of fits into the groove of actin. So all of that is tropomyosin. So again, how do we get this strong cross-bridge formation? Well, notice at the top that the troponin protein has a calcium binding site. So when calcium is present, it will bind to this troponin. Notice the way they've drawn this, and your book does this as well. Troponin sort of sits on top of tropomyosin. So they're connected with each other. When calcium comes in and binds to troponin, it actually sort of changes the shape of troponin. And since troponin is attached to tropomyosin, that will lift tropomyosin out of the groove of actin. And it lifts tropomyosin out of the groove of actin, and that actually exposes a binding site on actin for myosin. So sort of the job of tropomyosin is a blocker. It's blocking this binding site on actin for myosin. But if we can lift tropomyosin out of the groove of actin, that will open up this binding site, and this will allow a strong actinomyosin, a strong cross-bridge formation to occur. So a really nice diagram. Figure 1123 is a nice diagram in your textbook. They have a version that's both relaxed and contracted state, so that's actually gives us a little bit more information. So the next obvious question then is where does this calcium come from, and how can, because we need to get calcium to bind to troponin to lift this tropomyosin and prevent it from blocking. So the current model, it's an excitation contraction model. And so we're going to go backward and go to the beginning, more to the beginning of our book. And we're going to look at the neuromuscular junction. So this is where a motor end plate is coupled with muscle contraction. So a nerve impulse connects with the muscle cell at the neuromuscular junction. And this nerve impulse travels down those T, and T stands for transverse tubules. And we saw that in earlier notes and video about the structure of muscle. It travels down these transverse tubules, and somehow, it's not completely understood, opens up channels that allows calcium to be released from the SR. The SR stands for the sarcoplasmic reticulum. And once that calcium can be released, and then these second and third bullet points are what we previously talked about. This allows for calcium to bind to troponin, causes a position change or a change that will lift tropomyosin, exposing the active site on actin-4 myosin, and this permits strong binding of actin and myosin, and a cross-bridge formation can occur and a contraction can occur. So the nerve impulse goes down these transverse tubules and releases calcium. So the next question then is, how do we get this nerve impulse going down this transverse tubule? And this is at the neuromuscular junction. So this is where a nerve and a muscle cell will connect. And so this is a nice picture out of your, or similar picture out of your book, of figure 1118. But actually look at 11.8. But actually look at 11.8, 11.9, and 11.10. So all of those different figures are really good that describe this. So we're sort of going backwards actually. So here's a nerve coming in. They've magnified this version of it right here out of this little box. And so this is this box over here. And here's some mitochondria, a lot of mitochondria. And there's a little bit of a space in there that's called the neuromuscular junction. And this is the motor end plate. And this is actually the sarcolemma. So, and it's all this increased surface areas. There's all these folds. And the sarcolemma is the muscle cell membrane. And here's some mitochondria in the muscle cell. And notice, it's sort of hard to see, but notice in this picture are different synaptic vesicles. A vesicle is basically a ball. And these synaptic vesicles contain the neurotransmitter that's called acetylcholine. So they contain acetylcholine. And when a nerve impulse travels down this nerve, these synaptic vesicles will migrate to the end, the axon of this nerve. And these synaptic vesicles then fuse to the end or the membrane of the end of the nerve and release their contents out into this neuromuscular junction. And the contents are acetylcholine that will then bind to acetylcholine receptors on the neuromuscular junction, on this sarcolemma, on the muscle cell membrane. And that is then what causes the action potential from the nerve to go to this muscle cell membrane. And now we have an action potential that's traveling along this muscle cell membrane. And I think in my next slide, here's again a picture from our book. Here's the synaptic cleft, and so now we would get this muscle cell membrane to actually have an action potential that would be flowing along the membrane. Here are these synaptic vesicles, a little bit nicer picture, and they will release their contents into this synaptic cleft. And their contents are the neurotransmitter acetylcholine. So now this is a picture that I copied from a different book, but here at the top, so we can look at Number one, we have an action potential now that's going along this sarcolemma, along the muscle cell membrane, and it hits a transverse tubule. The transverse tubule dives deep inside the muscle cell, and gets really close to the sarcoplasmic reticulum. Somehow, it's not completely understood, but that then causes release of calcium out of this sarcoplasmic reticulum. And then you know that calcium will bind to troponin. Here they draw it as like a Pac-Man shape kind of thing. Troponin is sitting on top of tropomyosin, so it lifts tropomyosin out of the groove of actin and causes a strong binding between the myosin head and the actin protein, and a cross-bridge formation can occur. So a really nice description and some really nice figures out of our textbook. And actually, I would ask you to look at pages 352 and 353, and it's a great description of the flow chart, so a step-by-step process of how muscles contract. And they have a really nice picture, and I've copied it in the PDF file and some other notes that I've posted online. They have a really nice picture on page 353, it's figure 1124, of this whole process. So you should know this whole process, and it's certainly a possibility for a final exam question. So a really nice description of the current hypothesis. Still not completely understood, but this is probably what's going on in terms of muscle contraction.